Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about plant growth regulators or phytohormones. So these are simple molecules of diverse chemical composition which may be indole compounds, adenine derivatives or derivatives of the carotenoids. Classification based on their nature of action. Plant growth promoters are the auxins, gibberellins and the cytokinins, and plant growth inhibitors are the abscisic acid. Ethylene may fit in either of the two groups, but it is a inhibitor. Next, history, if you see, so for the first time, Charles Darwin and Francis Darwin discovered this auxins. The discovery of auxin dates back and while the Darwin was studying trophism in the plants. He exposed grass coleoptile to the unilateral light and observed it to bend towards the light. He covered the coleoptile tip with a thin foil or cut it off so that it was not acted upon by the light and observed that coleoptile did not bend towards the unilateral light. Thus, he concluded from his experiments that some stimulus is transmitted from upper to the lower part which induced bending of the coleoptile. So later, Boysen and the Jensen cut off the coleoptile tip and replaced it with a thin plate of gelatin inserted between the tip and the cut stump and observed that a coleoptile could still bend towards unilateral light, although he did not give any explanation. But it was evident that by the experiments of Darwin, stimulus was in fact a material substance which was in the control of growth. Next, isolated for the first time, auxin was isolated by F.W. Wendt from the tips of coleoptile of oat seedlings. So that is Avena coleoptile, and placed them on small agar blocks for certain periods of time and then placed the agar blocks asymmetrically on cut coleoptile. Later again they observed the growth of the plant and later he named it as auxin. Next, natural auxins are indole 3 acetic acid, indole butyric acid. Synthetic are 2,4-D, that is 2,4-dichlorophenoxyacid acid, naphthalene acetic acid. And tryptophan is the precursor for the auxin synthesis in the presence of zinc. Functions, cell elongation of stems and the roots. So this auxin helps in the growth, that is elongation of stems, roots will takes place, so growth will increase. Apical dominance, so indole acetic acid is in apical bud suppresses the growth of the lateral buds. If we remove that apex region, the lateral buds will start growing. It induces the parthenocarpy, which is the formation of a fruit without fertilization. So seeds are not produced here. So that was induced by the auxins. Examples in tomato. Actually, banana is a natural parthenocarpic fruit. It prevents premature fall of the leaves, flowers, fruits, useful in the stem cuttings and grafting, where it initiates the rooting process and also it will increase the stem growth. Promotes flowering in pineapple, mostly it will increase the female flowering. 2,4-D is widely used as a herbicide to kill the undesirable weeds of dicot plants without affecting the monocot plants. Helps in the cell division and xylem differentiation. So these are the important functions of the auxins. Please subscribe my channel if you like it. Thank you.